So right here is the MEA recorder, and what we do is we put a 48 well plate into it, and in each well of the plate there are about 16 electrodes, microelectrodes, that can detect cells basically communicating with each other on the plate, and this is the live action version of what we see is happening in the wells. So each of these spikes right here is basically a signal that we see popping up in the plates, and right here you can kind of see what activity is happening and where it's happening most prominently. And what we're kind of looking for is for some even activity to be happening, because that kind of shows that the cells are communicating at the same time. And what's happening is that the cells are creating extracellular field potentials, and when a field potential above 5.5 is picked up within a cell, that's when we start to see these little white spikes. And what we're going to be looking for in the end is comparing these network bursts, which are these red thick bars that happen along each of the 16 electrodes, which you can see horizontally. So these cells particularly have been exposed to sodium arsenite, and as you can see they're in this plate right here. And what we're going to be comparing is, right now these cells are about, they're about 149 days on this plate, and about 15 days ago we had exposed them to sodium arsenite, and what we're looking at right now is to pick up patterns on the recovery rate of sodium arsenite and how the sodium arsenite single exposure had affected the signaling pathways within these cells.